Welcome student. In last lecture we have seen about transcription. In transcription we come to the point that from DNA mRNA is formed and that mRNA copies information for various life processes or for protein synthesis which regulates various types of function in our body. So if we consider that DNA is a master molecule, DNA is a master molecule in living things because DNA has a genetic material which is transferred from one generation to the next generation, and the functions which has to be carried are also carried. And this DNA has an information copied by mRNA from DNA, and this DNA is made up of nucleotides, and this one nucleotide consists of one nitrogen base. And if we see the information for various processes, various functions is coded into codons, is coded into a triplet of nitrogen bases. 3 nitrogen bases comes together and form a triplet that is called as what? Codon. And this codon specifies certain amino acid, and these certain amino acids, when is carried to ribosomes and after translation forms a chain of polypeptide chain, and that protein is synthesis likewise. So this DNA is a master molecule, and if we see the site for information storage is in the sequence of nucleotide or in the sequence of nitrogen bases. And if we see the 20 amino acids which are called as essential amino acids, these essential amino acids are coded on nitrogen bases, on codons, triplet of nitrogen bases, that is called as what? Codon. So these 20 amino acids are encoded by 4 types of codon. By 4 types of, we are seeing the next point, that is called a genetic code. So these 4 nitrogen bases forms various types of codon. If these codons are in uniplate, this is a singlet, that is called as singlet form. Then it will form singlet form is only one, in one codon there is only one nitrogen base, one nitrogen base in one codon. And so this will form how many codons? 4 codons. Then there is also a doublet form. When 2 nitrogen bases comes together and form codon, then that is called as doublet. And that doublet consists of 2 nitrogen bases. And it will form 16 codons. And in living things, Moreover, the codons are in triplet form. Triplet form, it consists of 3 nitrogen bases and it will form near about 64 codons. And if you see living things or in human, in animals, there are possibility of 64 codons. 3 triplet form is there. 3 nitrogen bases comes together and form one codon. And this one codon specify only one amino acid. This one codon specifies for one amino acid. So if you see according to the 4 nitrogen bases, the 4 nitrogen bases forms near about 64 codon, and there is a dictionary given by Nirenberg, then Matty and Okia, the dictionary of genetic code, that how these codons are formed, how 64 codons are formed. If we see this possibility, I will explain in only one example, one tablet form, that how this dictionary is formed. If you see, there are near about like this 4 columns are there. Like this 4 columns are there. Here first letter. Here second letter. Here third letter. Third letter. U cat. U C. Here it is G, last one. Here first one U C E G. I will take this only one column. U C E G, like this. U A C G. Now what happens when codons are? This is a short form, or this is a very useful method for remembering the dictionary of genetic code, in which if we take the first letter here, this is the first letter. Now second letter, this column includes U. Then this third letter, first line, this carries U. So first codon is U. Then second, here in this column there are, in each column there are 4 codons. There are 4 codons. Now second codon, say first letter U, second letter here C, second letter U, here U, here third letter is C. This is second codon. Now third codon, first letter U, here second letter U, here third letter A. Then fourth codon, first letter U, then second letter U, and third letter here G. Likewise, 4 codons are formed. This is the simplest method. Now second column, first letter here U, second letter here C, first letter here U. Then first letter U, second letter C, third letter C. Then first letter U, second letter C, third letter U. First letter U, second letter C, and third letter G. So if you see, first letter is common in both the columns here. Second letter is also common from the upper one. Here second letter is common. Here C. Only what changes? UCAG. This you have to write it. Now how you will write this? First letter U. Second letter A. Third UCG. Now fourth column, first letter U. Second letter G. Third letter UCGA. Like this genetic code dictionary is to be written. Now what is genetic code? It is a cryptogram. It is a cryptogram or it is a coded language for protein synthesis. Which is copied by mRNA from DNA. And this mRNA transfers the information for protein synthesis to ribosome, and with the help of ribosome and tRNA, the polypeptide chain is synthesized or protein is synthesized. So actually it is a what? It is a information copy. It is a information for protein synthesis. It is the information for protein synthesis, and this program specifies certain amino acids. This program specifies certain amino acids. So, genetic code. This is our genetic code. We will write like that. So, genetic code is a collection of base sequences that specify particular amino acids. In all amino acids, we see that a polypeptide chain of amino acid form proteins. In short, synthesis of protein, this information is coded into genetic code, and this genetic code, or it is simply what? It is a sequence of nucleotides. It is a sequence of nucleotide or nitrogen bases. 3 nitrogen bases, that is a triplet which forms, this triplet from codons, and this specify specific amino acid. So now we will see the characteristics of genetic code. Characteristics of genetic code. This question is asked in the examination, board exams, and is very important for NEET exam and also CET exam. So, if we see the first characteristic, genetic code is a triplet code. Genetic code is a triplet code. Now, if you see, there are 3 consecutive bases constitute a codon. 3 consecutive, as we see in RNA. These 3 consecutive base sequences is A, U, G. This is what? Adenine nucleotide or adenine nitrogen base, vinyl nitrogen base. This is called as what? Codon. And this codon specify one amino acid, that is, suppose AUG is specify methanol. Means genetic code is in a triplet form. It is a triplet code. So it specifies certain amino acid or only one amino acid. Then all living organisms have a triplet code. In all living organisms, the codon is in triplet form, and this triplet codon specifies certain amino acid. Then second characteristic, genetic code has a distinct polarity. Genetic code has distinct polarity. Now if you see, it shows, genetic code shows definite polarity, that is definite direction. If you see DNA, this is 5 dash, this is 3 dash, this is 3 dash, this is 5 dash. Direction, always the direction is in 5 to 3 dash. This is 5 to 3, anti-parallel. Where both the strands are anti-parallel. So polarity or direction is in a definite. It has genetic code has a definite polarity, and you always read in 5 prime to 3 prime. Always read into 5 prime to 3 prime direction. So genetic code has a specific polarity. Then genetic code is non-overlapping. Genetic code is non-overlapping. If you see, each single base is a part of only one codon. Each single base is part of only one codon. Adjacent codon do not overlap each other. Adjacent codon do not overlap each other. So there is no overlapping of adjacent codons. Then fourth point, commaless. Genetic code is commaless. No gap or no punctuation, comma. So there is no gap between two consecutive codons. There is no gap. They are continuous in nature. If you see, if this is a mRNA, this is first codon, this is second codon, this is third codon, there is no gap. There is a connection between this and there is no gap. So it is commaless. Genetic code is commaless. Now we will see fifth point. Genetic code has degeneracy. Genetic code has degeneracy. So if you see, single amino acid is encoded by single codon, and say some amino acids are encoded by more than one codon. Now if you see, this specify only one amino acid. But sometimes what happens? Two types of codons may specify same amino acid. Same amino acid is specified by two different types of codon. So that is called as what? Degeneracy. That is called as what? Degeneracy. If we see cysteine, two codons specify cysteine. Example, cysteine is carried by two codons. Then, for example, isoleucine is carried by three codons. In all, how many codons are there? 64 codons are there. And these codons specify 20 different amino acids. So, many codons specify same type of amino acid. This is called as what? Degeneracy. This is called as what? Degeneracy. Then, if you see the next characteristic, genetic code is universal. Genetic code is universal. In all living things that has genetic code, in that living organism, suppose AUG, this codon is present. This codon always in all organisms will specify methionine. It will not specify aspartic acid in other or methionine in one. In all living organisms, if this codon is present, it will specify methionine. So, this is universal. Genetic code is universal. It is obeyed by all organisms. Then, genetic code is non-ambiguous. Genetic code is non-ambiguous. Specific amino acids encoded by a particular codon. Two different amino acids never be encoded by the same codon. So, if we see one codon specifies only one type of amino acid. Only one type of amino acid. It will not, if suppose this is one codon, it's AUG. If it specifies methionine, it will not specify isoleucine. It will specify only methionine. Two different amino acids are not specified by one codon. This is called as what? Non-ambiguous nature. This is called as what? Non-ambiguous nature. Then, 8th point. Initiation codon and termination codon. Initiation codon, from which AUG, this codon is initiation codon, from which the reading of genetic code started. And if you see, and it is present in 5' direction. It is present in 5' point. It starts from 5' side. And if you see stop codon, UAG, UGA and UUA. This, for example, this I have given 2 only, but there are more than 2 means 3 stop codons are there. These are stop codons at which at this site the proteins at this site the chain of polypeptide chain ends here. Stop codon. So reading stops here, and reading stop means there is the end of polypeptide chain here. So these are called as what? Stop codon. Then codon and anticodon. Last point, codon and anticodon. If you see, codon is present on DNA also, mRNA also, and anticodon is present on tRNA. Anticodon is present on tRNA. And it is specified, it is represented by 3' direction to 5' direction UAC. 5' direction. And if you see codon, 5' AUG to 3' AUG. This is called as what? Codon. And this is called as what? Anticodon. This is called as what? Anticodon. So these are the characteristics of genetic code, and these are always asked in the exam. So next point we will see in next period.